Hello, everyone, and welcome to today's ASHI Educational Initiatives Committee webinar, Basics of HLA and Transplantation. We are delighted that you have joined us for today's educational session. My name is Kristina Olack, and I'm going to be serving as your moderator today. Before we begin, I'm going to go over just a few housekeeping items. Today's webinar is recorded and is going to be available later today on ASHI University and ASHI's YouTube channel. At the end of this week, you'll also receive an email containing a link to complete a brief evaluation. And once that evaluation is submitted, you will receive your continuing education certificate through the email. Throughout today's presentation, please submit any questions you may have using the chat feature, and we will address as many questions as possible in the Q&A session at the very end. Right now, it is my pleasure to introduce today's speaker, Dr. Lalli. Dr. Lalli is the director of the HLA and flow cytometry laboratory here at CMC in Charlotte, North Carolina. He also serves as the director for histocompatibility laboratory at Lehigh Valley Hospital in Allentown, Pennsylvania, as well as Aurora St. Luke's Medical Center in Milwaukee. In addition to his clinical roles, Dr. Lalli is also the past chair of the UNOS OPTN Histocompatibility Committee and has served on the MPSC, POC, and kidney committees. Please join me in welcoming Dr. Peter Lalli.
Thank you, Kristina. And welcome, everyone. Today, we're going to kind of go over the basics of what HLA is and its role in transplant rejection, and then a little bit about how the histocompatibility laboratory supports our transplant programs on the clinical side. So no disclosures for this presentation. So to start with everything, back to your basics of immunology, anything that is not part of you is bad and needs to go away as far as the immune system is concerned. And that's what we're going to focus on today in kind of understanding the concepts of how transplant immunology works and how HLA plays a role in that. So there's a couple of key terms that we talk about when we're talking about how the immune system recognizes whatever's going on in the world. So we have the concept of self. So self is anything that's normally part of you, what your body's made of. Your immune system looks at that and says, okay, this is okay. Then we have non-self. So that's anything else that the body doesn't recognize, anything that's not normally part of you. The immune system is kind of targeted to say, this is bad, something's going wrong here, and we need to do something about it. The last one is something we call altered self. So these are normal molecules in the body that somehow change for one reason or another, and or mutated from normal. So when we think about this, we normally think about tumors. So if you have a mutation that leads to tumor-producing protein, those proteins are going to be different than what your body's normally used to, which allows us to have an anti-tumor type immune response. So if we go back to basic immunology, we have a couple of arms of our immune system. We have our innate immune system, or our natural and native type of immunity. And in this case, it's less evolved. It's more for a first line of defense. So anything that may enter the body, your innate immune system is the first to respond and try and clear out whatever may be harmful to your body. On the other side, we have the adaptive response. The adaptive response is a specific response that is more advanced. It's more tailored to whatever the stimulus is that's going on here. It does take a little bit longer to develop initially, but is much more effective and ends up having stronger responses with each different exposure, with the ability to remember previous infections or pathogens. So we talk about what happens in the immune system. We have our innate immune response here early on in the time frame after exposure. We have things like our phagocytes, our epithelial barriers. We have complement proteins that can attach to microbes and kill them off. And natural killer cells can kind of go in and start attacking whatever may be insulting the body. And as time goes on, our adaptive immune response kicks in with our B lymphocytes turning into plasma cells and making antibodies, and our T lymphocytes increasing, turning on killer T cells that can then go and either kill off virally infected cells, or in the case of tumors, it can kill off tumor cells as well. So I bring all this up to kind of remind you of how the immune system thinks about things. And in our case, why is our immune system able to recognize another person's kidney that we put in as a transplant? Why does it think it's wrong? And what does it try to do about it? So as I said before, the immune system is trained to recognize self. So everything that makes up you, your immune system is able to recognize and say, okay, I know what this is. This is fine. I'm going to leave it alone. But then anything that is non-self, anything that your T cells or your B cells don't recognize, it considers foreign and potentially dangerous. So these can be things like bacterial sugars, viral proteins, and more importantly for this presentation, variations of human proteins that are not normally found in a given individual. So in other words, any person's HLA proteins that aren't yours, your immune system is going to recognize as foreign and try to fight it off, thinking that it's bad, even though we all know that it's not. So when we talk about what happens in transplant rejection, this is a great kind of early on experiments that you can do where you take a mouse, you take a piece of skin from one strain of mouse and you graft it on to the same strain of mouse. So all of the proteins in this mouse are the same, in the donor mouse are the same as the recipient. So in this case, the immune cells look at this and say, okay, I recognize this. This looks fine. They don't do anything to it. They just leave it alone, and then that graft can heal in and not get rejected. But if we take the graft, the skin graft from one strain of mouse and put it onto a different strain of mouse where the proteins and the MHC proteins are different, this mouse is going to reject it. It's going to say, I don't know what this is. It must be bad. We need to kill it off. So if you do the same experiment where you take a skin graft from a B strain of mouse and then put it onto a mouse that is an F1 progeny of an A and B. So this mouse inside of it has both A and B proteins in it. This graft is going to be accepted because it looks at the B from the donor and says, okay, this is fine. But if we flip it around and use the mix of A, B and use that as the graft donor onto an A only, this mouse immune system is going to recognize that B protein as being foreign and kill off that graft. So this is just a experimental way of saying that anytime our immune system sees something that it doesn't recognize, it's going to try and attack it. So when we talk about humans, the proteins that we're discussing in these cases are called HLA, and HLA stands for human leukocyte antigen. It's a group of 6 different proteins or 6 different gene products. We have class 1. Class 1 is found on all nucleated cells. So by nucleated cells, we mean anything in the body except for red blood cells. There's a couple other minor exceptions to that, but by and large, all of the cells in your body are going to express these class 1 proteins, including epithelial cells, endothelial cells inside of the blood vessels in a kidney graft, heart muscle tissue. All of those things are going to express these class 1 proteins. We are scientists, obviously. We're not incredibly creative, so we named them A, B, and C initially on the class 1 side. Class 2, on the other hand, are found on only specialized immune cells. So these are going to be all of the antigen-presenting cells that we have in our body, like our B cells, dendritic cells, monocytes, macrophage. The jobs of these cells are to go around and sample the environment, take pieces of them, and show them to the T cells that we have. And in those cases, they're shown through these class 2 molecules. And again, we initially called these D, and then we realized that there were more of them, and they were split into smaller categories. So we have HLA-DR, DQ, and DP. Now, one thing to note, and we'll see this in a second, is that all of these HLA genes are really close together, including being close to a lot of these different immune inflammatory response genes. So if you have a circumstance where you have inflammation, cells that don't normally express these class 2 proteins can turn on these proteins in cells in the case of inflammation. So the reason I bring this up is the whole transplant process is incredibly inflammatory. So you're taking an organ from a person that is somewhere in the process of dying. You cut off the blood supply. You have a bunch of surgical trauma while the surgeons are trying to take the organ out. You put it into an ice bucket or onto a pump. Then you take that back in, put it in there, have all of this new blood rush in. That whole process is really, really inflammatory. So during the transplant process, it makes it so these HLA class 2 targets are present when normally they might not be. So just an important thing to think about during that transplant process, that we can have these targets that we might not have under normal circumstances. So HLA is highly polymorphic. It's actually the most polymorphic gene in the entire human genome. So if you look at genes like insulin from one person to another, they're almost exactly the same DNA sequence from me to you to anybody else. But if we look at HLA, they're all vastly different, and everybody is going to have some slight variation in their HLA typing. And these HLA proteins serve as a communication mechanism between immune cells. So it allows your cells to distinguish good from bad, or if we're talking more in the immunological sense, self and non-self. So this is what HLA proteins look like. The way I like to think about it is they look like little arms and hands that stick off from the outside of the cell. And inside of the hand, the HLA proteins will take a piece of its environment and show it to T cells. So the way these proteins work is they allow our T cells to see what's going on in their environment. So inside the peptide binding groove, or the little hand that we have on our HLA protein, they'll hold pieces of the environment out. Now, if you're healthy, it'll hold pieces of you out to your T cells, and the T cells will walk by it and say, yep, that looks fine to me, we're good. But if you have an infection or if you've had a transplant, it'll take pieces of other proteins, chop those up, and hold them out to your immune cells, your T cells, to allow those T cells to say, yes, this is good, or in cases of infection or transplant, it'll say, oh, something's not right about that. I don't know what that is. We need to try and fight that off. So these HLA proteins, their job is, as I said, to interact with the T cell receptor on T cells. So T cells can't just see their environment. The only way they can identify what's going on is by this interaction here. So these HLA proteins will interact with the T cell receptor. And under normal circumstance, what they're looking for is that little piece of peptide that the MHC, or the HLA protein, is holding to try and allow these T cells to identify, is this self or is this non-self? And if it's non-self, they need to figure out how to induce some sort of response to try and keep the body healthy. So in the case of transplant and in allorecognition, what's happening is a kind of a combination of seeing not only the peptide as being a foreign protein, but it also sees that whole HLA protein as potentially being different as well. So if we look to see how allorecognition actually works, we can look at how the T cell receptor sees what it's looking at. So there's two different spots on the T cell receptor that are trying to look at the HLA peptide complex. So we have the peptide from either your normal self proteins or from a viral peptide or a bacterial protein, as well as the normal HLA protein. So it's looking at what's in here as well as what's out here. So in the case of allorecognition, there's a couple of different things that can happen. So the T cell receptor may see an allogeneic peptide, so a peptide that it doesn't recognize from the donor's HLA molecules, or it can look at spots on here where it says, okay, I see part of the donor HLA protein, but I don't know what that is. I don't recognize it. So in this case, it'll activate and start to try and turn on a response to clear out whatever it thinks this bad protein happens to be. So when we talk about what the HLA genes look like, HLA genes are found on the short arm of chromosome 6. We have 3 different classes. I mentioned 2 before. Class 1 is going to be your A, B, and C proteins. Your class 2 are DP, DQ, and DR. And then class 3 contains a lot of other immune response genes like tumor necrosis factor. Some of the complement proteins are all encoded in this class 3 region of the HLA complex. And in the end, it gives rise to that arm and hand shaped protein that we have here. So in the HLA genes for class 1, we have 8 exons in our gene. We have the signal peptide. Then we have exon 2, 3, and 4, which make up the extracellular portion of the molecule. So alpha 1 is created by exon 2. Exon 3 is creates the alpha 2 domain, and then exon 4 creates the alpha 3 domain. Exon 5 creates the transmembrane, and then 6, 7, and 8 create the cytoplasmic tail. And then beta 2 microglobulin, which is a non-polymorphic protein, is made on another chromosome, gets produced, and then associates with this. And basically, it's to help hold the hand into place. It kind of is a structural protein that allows the molecule to stay in place and hold the peptide that we want to present to the T cells. So if we look at our HLA type, the way we started identifying these HLA proteins was using serologic-based methods, so taking anti-sera that we've gotten from patients or from placentas, from women that had previously given birth. We identified antibodies that separated out the different HLA proteins into groups. And we would go through and identify these groups, and as time went on and we got better antibodies that were more specific for one protein versus the other, it allowed us to separate these HLA proteins into what we kind of call groups at this point. So there's about 20 different HLA-A proteins, about 40 B, 9 or so C, 14 DR, about 7 DQs. You get 2 copies of each one of these HLA proteins. So 2 A's, 2 B's, 2 C's, 2 DR's, 2 DQs. One from mom, one from dad. And interestingly enough, if you look at somebody's HLA type, you can use it to identify parentage. So you can see if the potential man is the father of a baby. Each one of our HLA types associate into what we call a haplotype. So a haplotype is one of the chromosomes that we get from each of our parents. So we have your dad, we have your mom, and then from each one of them, we take one of the 2 haplotypes and mix them together to create the children. So as we showed before on one of the previous slides, all of the HLA proteins are right next to each other on the same chromosome, which means they get passed down genetically all together. And that whole group of all of your individual HLA proteins is what we call the haplotype. So in here, we can take that one haplotype from mom, one haplotype from dad, and create 4 different potential combinations here in the children. It's basically going back to your basic biology in high school and college where we were doing Mendelian genetics, Punnett squares. It certainly applies to how we get our HLA haplotypes from our parents. This is really important when we start looking at stem cell transplants. It allows us to identify which of our potential siblings, parents, or children might be suitable donors for us. So if we look at kind of what a basic HLA typing looks like, when we are in our laboratory, one of the first things that we do for all of our patients and all of our donors is identify what their HLA type is. Each one of us has a unique individual typing. And you can see here in this case, this person is HLA A2 and A3, HLA B27 and 35, CW2 and 4, DR8, 11, DQ7 and 7. So this is what an HLA type will look like. This happens to be my HLA type because it's the one that I remember when I'm making my presentations. As I said before, everybody has different HLA types. There's all sorts of different combinations of amino acids that can come together to form these HLA proteins. And if you look at all of the different variability that we have in different HLA proteins, if you look at the genes and look at each one of the individual amino acid residues, you'll see that in the alpha 1 and alpha 2 domains, which are the 2 outside portions of that arm and hand situation, where the T cell receptors are most going to be interacting with not only the peptide, but also the HLA protein together, that's where we find the bulk of the variability in these proteins. Alpha 3, remember, was the structural portion of the molecule, so we don't need much variation there. But by having all of this different variation, it allows us to have, as a species, a whole bunch of different HLA proteins to allow for us to fight off different infections. So if we look at it on kind of the global keep humans alive on the planet kind of situation, we want a ton of variation so that some population is always going to be able to survive some sort of infection or pathogen or whatever it happens to be, so that we're able to fight that off. And if you look at that from a genetic standpoint, we have all of this variation in our HLA genes that lead to a bunch of different proteins so that we have all of this variation among our species. So when we talk about HLA typing, there's two different concepts that we look at. So I mentioned before that we have kind of these general HLA protein groups. Well, when we look at those, we can break them down into what we call alleles. And these are going to be the different genetic sequences that lead to the final protein that we have. So there are many gene sequences that can lead to very similar proteins. And before we did our HLA or our DNA-based testing, as I said, the HLA typing was performed using these antisera that grouped the HLA proteins. Well, now that we've been able to use DNA, and now even further, now that we're using next generation sequencing for virtually all of our HLA typing, the DNA methods can identify many, many more genes within a specific protein group. So if we look at those protein groups that we talked about before, but now look at the genes themselves, we can see that there's a lot more variation in them. So if we talk about serology here, we're talking about something like HLA-A2. It's the most basic form of typing. It's this protein group that we can look at to kind of understand what the physical shape of that protein ends up being after that gene is transcribed and translated and turned into a protein. If we look at the molecular gene, though, we can see that there are many different types of alleles that fall within the HLA-A2 grouping. So if we look at the HLA-A2 protein group, we've identified at least 1,000 different variations of genes that all lead to this similar HLA-A2 protein. And if we look at how different some of these alleles may be, one good example is this HLA-B44. So B44 has two primary different alleles, 4402 and 4403. And if we look at the amino acid sequence for both of these proteins, we'll notice the only difference is this one amino acid difference between 4402 and 4403. And interestingly enough, our immune system is so sensitive that if you are a 4402, you can make an antibody or an immune response against 4403, even though the only single thing different between those two proteins is this one amino acid. That's enough for your immune response to look at that and say, this is not self. I don't know what this is. We have to do something about it. So just something really kind of cool about how sensitive and how able our immune system is to identify even minute differences between HLA proteins. So if we look at how many different HLA genes there are, as I said before, there were only a few different protein groups, but if we look at the number of alleles, there are tens of thousands of different HLA alleles that have been identified so far, and there's probably many more that we haven't identified yet. So almost 10,000 different A, B, and C alleles, 3,000 to 5,000 class 2 alleles. So there is a ton of variation in these proteins that allows us to, like I said, be able to fight off whatever nature may throw at our bodies. Not all of these frequencies are created the same. Some are more common than others. So for example, HLA-A2 is in about 46 to 48% of the US population, A11 in about 10%, and A80 is something that you may only see in a fraction of a percent of the US population. And interestingly enough, these HLA frequencies, the frequencies of what you may have, vary depending on where your ancestry is or where in the world your family grew up. And a lot of that has to do with the function of these proteins. Remember, I said that their purpose was to show pieces of the environment or pathogens that may be affecting an individual to the immune system. Well, if you have an HLA protein and a pathogen happens to fit very well in there and able to turn on your T cells, those T cells are going to be able to activate and clear those different infections so that the people that have that infection and that HLA protein are able to survive. And if they don't, then they may be more susceptible to having the pathogen kill them off. So if we look at the different HLA proteins around the world, remember we have different endemic pathogens all around the world that may affect different populations depending on where they happen to be. So one of the great examples that all of us kind of use in the HLA world is HLA-B46. If I see HLA-B46 on one of my patients, I know that they have Asian ancestry in their heritage. And the likelihood is that as humans evolved in the Southeast Asia region, there was a pathogen there that fit very well into that B46 protein. And as a result, those patients or those people that grew up in that area with that pathogen were able to fight it off and survive. Somebody that didn't have that kind of wasn't able to procreate and survive long enough to pass on their genes, as we see in general Darwinian evolution or natural selection. This is a great example of how that works. So whatever this pathogen was in Southeast Asia wasn't found in Europe, so we don't see B46 in Caucasian European people. Instead, we have something like AO3 that was common in Europe that fought off some sort of different pathogen. So it's really neat that you can go through and look at a specific typing and be able to trace the ancestry based on these different HLA proteins. Okay, so now we've talked a little bit about what HLA is, how it works. Let's talk a little bit about how it influences our transplants and our transplant patients. So when we talk about the immune system trying to fight off a transplanted organ, what we're talking about is a term called immunologic rejection. And immunologic rejection is the targeting of the transplanted organ by components of the immune system, including cells and antibodies. So HLA can be targeted by both a cellular mechanism where the T cells turn on and our cytotoxic T cells will go in and try and kill off the graft cells, or through an antibody-mediated mechanism where the B cells turn into plasma cells and make antibodies that will then stick to the HLA proteins that are on the donor cells and cause cell death in those cases. So antibodies in the laboratory are what we care about most, or what we deal with most. And they're the primary effector mechanisms of B cells. They are these basically little Y-shaped proteins that have a constant region that's the same from antibody to antibody. And then they have this variable or antibody binding region up at the top. That's kind of the business end of the molecule. This is what sticks to whatever pathogens we have, viruses, bacteria, HLA proteins. They stick and they cause downstream effects that can lead to the death of the pathogen or to the donor-derived cells. So the way this works is kind of in the most basic sense, the antibodies stick to the HLA protein, then a molecule called C1 will come in and attach, and this C1 interaction here initiates a cascade using other complement proteins that in the end leads to this cell death by creating what we call this membrane attack complex. Basically, it's a way for the immune system to poke a hole in the cells and cause the cell death of those of the cell that's attached to it, and make it so that that cell will end up dying. Well, I always point this out because in the histology lab, when we do a biopsy on one of our patients, we want to be able to see what's going on in there. And one of the tests that we perform in there is to look to see if our patients have antibody-mediated rejection or antibody-mediated injury. So when we do a biopsy, we take the tissue out, we put it through the whole histology process, and then we look at it under the microscope. And when we're looking at it, we have a couple of tools in the laboratory that allow us to see what's going on. Well, unfortunately, what we really want to see is, are there antibodies that are sticking to our graft cells? And unfortunately, the interaction between the antibody and the HLA proteins is actually a pretty weak interaction. So as we go through and do the histology process, the sectioning, the fixing, the staining, those antibodies will fall off and we can't see them no matter how hard we try. So what we do instead is during this process, one of the complement proteins, particularly the C4 molecule, covalently binds to molecules that are on the surface of the donor cell. And that covalent bonding allows this protein to stick around even through the whole histology process. So instead of looking at an antibody on our histology, what we look for is this C4D molecule. It's basically a shadow that was left behind from having the antibody stuck. So if we have a graft and we do this histology staining and we see that there's a presence of this C4D molecule, it tells us that at some point in time there was an antibody stuck there, and that's a signal to us that this patient is undergoing antibody-mediated rejection. So if you ever see a histology report when we're looking at a post-transplant biopsy and they comment on C4D, this is what they're looking at. They're trying to see, was there an antibody there at some point? All right, so we have a couple of different types of rejection that can occur in a transplant situation. First off, we have what's called hyperacute rejection, acute rejection, and then chronic rejection. So to kind of quickly go through the three types, hyperacute rejection happens very quickly after transplant, within minutes to hours to days after we put a graft in. And in these cases, this is due to a preformed immune response that has already occurred for some reason or another. So if you've already been exposed to someone else's HLA proteins, your immune system will create antibodies that are going to stick around for the life of your existence. Those antibodies are constantly going to be floating around in your serum. And if we take a kidney and put it in, and that donor happened to have an HLA protein on the surface of their cells that you had already been exposed to as a patient and have created antibodies, as soon as we put that graft into your body and release the clamps and allow that blood to flow through it, those antibodies that you have circulating are going to see all of these different donor HLA proteins inside of the blood vessels and start attacking it immediately. And that's what happens when you have hyperacute rejection. So what happens is these antibodies stick to the endothelial lining of the donor blood vessels, and it causes this immediate reaction where the cells get attacked, they start lysing. That lysis will lead to complement activation, endothelial cell damage, and eventually inflammation and thrombosis. So when we have hyperacute rejection, what's actually happening is this immune response leads to a widespread thrombosis, and the blood inside of the donor organ just ends up clotting and kind of chokes off all the blood flow and kind of makes it so that the organ becomes devoid of oxygen and nutrients and can die off fairly quickly. So ideally, this is not what we want to see. So where does this preformed alloreactive memory come from? Well, there's a couple of different ways that this can happen. First off, if you've ever been transplanted before, you've been exposed to someone else's HLA proteins. And in those patients, those that we see in the laboratory that are the most sensitized generally are those that have previously been transplanted. So it kind of acts as a vaccine for our immune system so that they've already recognized these proteins. And as I said before, the adaptive response every other time you see something gets worse and worse. So what we see is that when we have a patient that's previously been exposed to these HLA proteins, their response, their secondary response is going to be a lot stronger. So you can have it through transplantation. If you get blood transfusions, you can have HLA proteins get carried over through that transfusion. And then pregnancy is also another very common way that somebody can become sensitized. So if you remember, the immune system recognizes anything that's not normally part of it as being foreign. Well, if you look at the fetus, half of the fetus comes from the father. So there are half of the HLA proteins in that baby that are not native to the maternal immune system. So the maternal immune system can actually recognize these paternal HLA proteins as being foreign and make antibodies against them so that if they're exposed to those same HLA proteins later on, it can create a response and lead to something like hyperacute rejection. Acute rejection, on the other hand, generally happens after the first week post-transplant, but can happen at any point in the life of the graft. And that's why we have to treat our patients with lifelong immunosuppression, because at any point in time, the cells of the immune system can kick on, recognize this as being bad, and start to try and fight it off. The same way that they try and fight off a cold or a scratch that you may get infected, anything that might influence your immune system to turn on, it can do in the same case when we talk about acute rejection. So in these cases, it can be a cellular or humoral response. It's one of the most significant hurdles that we have in the maintenance of our grafts. So we are constantly monitoring our patients through a bunch of different mechanisms to see if this may be going on. If we catch it early enough, we may be able to intervene, treat the rejection so that we don't lose too much graft function. But in some cases, if you have a patient who happens to stop taking their immunosuppression for even sometimes as short as a week, it can lead to the immune response kicking on so badly that it destroys the graft, and we may lose the graft in cases like that. So in these cases, the cells can start attacking. They can make antibodies that can stick to the HLA proteins on the donor cells as well, in the end, all leading to some sort of damage to the tissue itself. And with damage of tissue leads to loss of function in the graft. Chronic rejection, on the other hand, occurs gradually over the life of the graft. It happens in almost everybody to some degree. It can be caused by both cellular and humoral mechanisms. And this is the one that we know kind of the least about. There's a bunch of different things that can happen over time as this goes on, but the idea is that it's just kind of a slow burn reaction that's going on. So the way I like to think about it is it's kind of the immune system bullying the transplanted endothelial cells inside the blood vessel. So every day, these immune cells walk past, they look at the HLA proteins that are on the graft cells and say, I don't recognize this. They're not so angry that they really try and kill it off, but every day they walk past and just kind of give it a little punch. And over time, that stress of being bullied over and over again, the cells of the immune system, or the cells of the graft, will then start to do what we all want to do when we're stressed, and that's make copies of ourselves. So when we have chronic rejection, what happens is this low-level reaction causes the cells inside of the blood vessels of the transplanted organ to start dividing and replicating. And over time, this replication leads to fibrosis and scarring inside of the blood vessel. And what happens is the outside of the blood vessel isn't going to stretch and get bigger. So as these cells divide, the inside of the blood vessel will start getting smaller and smaller and smaller. So a blood vessel that used to be this big is now this big, and in some cases can completely be occluded by this scar tissue. If you have occlusion of these blood vessels, you lose blood flow to the organ, and over time, it can actually lessen the function of whatever you happen to be. So if we look under the microscope, this is really what we see. So this elastin staining looks at the inside of what the blood vessel used to look like. And now you can see all of this stuff has kind of crept in over time and made it so that this blood vessel is significantly occluded compared to what it was when we first put that organ into place. And this can happen over you can have accelerated chronic rejection that can happen in a year or two. And there are some patients that have had grafts for 40 years and have very little of this chronic rejection. It's very dependent on patient to patient, and something that we at this point don't really know the best way to treat. All right, so we talked a little bit about what rejection looks like. Let's talk a little bit about how we can prevent it. So we have two different ways that we can manage this kind of stuff in the clinic and in the laboratory. The first is going to be immunosuppression. All of our patients get lifelong immunosuppression, and we use this to try and keep the immune system from getting so angry that they try and fight off the HLA proteins that are in the graft. We need to be able to balance this so that they can fight off infections and keep patients healthy from foreign invaders, but not get so angry and try and fight off the actual graft itself. So in the clinic, we treat with immunosuppression. The other half of the equation is what we in the laboratory are able to do to help provide information to our clinicians to perform the safest possible transplants that we can. And the way we do this generally is by screening the recipient serum for the presence of antibodies against the donor HLA. Ideally, we would test for antibodies and cellular responses, but unfortunately, the tests that we've been able to develop over the years have never really been able to identify or grade the cellular response against a potential donor cell. So at this point, our best tools are to look at the antibody response in our patients. So if we go back to the late 1960s, we were able to identify that anti-HLA antibodies in transplant were incredibly important for the survival of a graft. So this goes back to Dr. Patel and Dr. Terasaki and their kind of seminal paper that said, okay, if we have patients that we have a positive cross-match, and by cross-match, what I mean is taking cells from the donor, antibody from the recipient, mixing them together and seeing if that recipient has antibodies that stick to the donor cells. If they do, that's what's called a positive cross-match. If they don't, that's a negative cross-match. And what they were able to find is that when they looked at patients that had a positive cross-match before transplant, those grafts failed immediately or within at least the first 3 months. However, if the patient had a negative cross-match, so no antibodies against that donor, their grafts survived. So when we look at the numbers here, it's really staggering to see that if you have antibodies against your donor, you're probably not going to have that graft survive. If you don't have antibodies, you have a much better chance of allowing that graft to survive for a long enough period of time. So this was incredibly important to us. It told us that before every transplant that we do, we need to ensure that this recipient doesn't have antibodies against this donor's cells, or else the graft is not going to be able to survive. So at that point, we developed an assay called the PRA. And the PRA was a test that allowed us to look at how likely it was for our patient to have antibody against a specific donor. So what we're able to do here in a screening type way was to take a whole bunch of, we'll say, mock donors. So these are going to be volunteers that we have in the laboratory, in the clinic, and we were able to draw cells on them and put different cells in each one of these wells on a tray. And then what we did was we took the patient serum and put it in all of the wells. So really what we were doing is taking 50 different cross matches all at the same time, and then seeing what the likelihood was that our recipient was going to have a positive cross match against any potential donor that's out there. So if we look at this case, we have 50 different donor cells, we mix it with our recipient serum, and we see that 10 of those donor cells caused a reaction with our patient's antibodies. So what that tells us is this patient had a 20% likelihood that they were going to have a positive cross-match. So what that said to us then is the lower the number, the more likely it was that a graft for a potential recipient was going to be okay for them. So a low number is good, a high number is bad. The higher the percentage, the more likely it is that this recipient is going to have a positive cross-match against any given donor that's out there. So in this case, a 20% PRA lead to a 1 out of every 5 donors would be unacceptable for them. Well, those assays were cumbersome. Dealing with live cells was kind of a pain in the butt. So we developed new assays that were able to allow us to look at this in a slightly different fashion. And that was the time that an instrument called the flow cytometer came about. And in this case, the flow cytometer is able to look at not only whether or not antibodies are sticking to cells, but it allowed us to take plastic beads and coat those plastic beads with recombinant HLA proteins and see whether or not the patient's antibodies stuck to these donor or to the HLA proteins that we put on these beads. So the way this works is you have your patient's serum, you mix in either a cell or a plastic bead that has HLA proteins on them. If the patient's antibodies that are floating in the serum recognize these specific HLA proteins, they'll come in, they'll stick, and then through the detection process in the flow cytometer, we can see that these antibodies, we can put these fluorescent tags on them so that the more antibody that's sticking to these cells, the more of these fluorochromes stick and the brighter the signal from the instrument gets. So if we look at this in how the instrument reports things, the y-axis, or I'm sorry, the x-axis looks at how much antibody has stuck to these cells, or how bright the fluorochromes are. And the fluorochrome amount is dependent upon how much antibody there is. So if you have no antibody, there's very little signal that comes off of these. If you have a lot of antibody, there's a lot of light that will come off of here. And this continuum shows us how much antibody our patient has against any given protein. And this number that we have on the x-axis is what we call the mean fluorescent intensity, or MFI. You'll hear us talking about this in the laboratory sometimes. The MFI basically is just a measure of how much antibody there is. So what we do in the laboratory is we're able to take these beads and mix them with our patient's serum. And if those antibodies stick, we get the signal in the flow cytometer. So the more antibody that sticks, the more signal that we get. So if we look at, this was kind of the old way that we used to do this on the flow cytometer, each individual grouping here is a different bead. And on each one of the beads, we have a different HLA protein. And since we know which bead has which HLA protein on it, when we mix it with our patient's serum and we see a positive reaction, so when they move over to the right, remember that means antibody has stuck, and those cells or the beads are now glowing brighter because of that, we can see not only which of those beads have antibody stuck to them, but based on how far they move, it gives us an idea of how much antibody there is. So if we look at this patient here, you can see that these three beads have all moved over to the right because they have patient antibody stuck to them. And we're able to quantify this difference on our, we don't really see these plots anymore. We just have a readout of these graphs like here. The y-axis on here is now that MFI value of each one of these different reactions. So the higher the bar, the brighter the bead, the more antibody there is. And now each one of these bars represents a different HLA protein. And we can go through and do this in a screening method on all of our patients before they go to transplant. So this gives us information about what's going on with our patient's antibodies at any given time. Now, it's important to note that inside of our serum, we have tons and tons of different antibodies. Not only do we potentially have anti-HLA antibodies, but we have anti-flu, anti-COVID, all sorts of different things. And one of the things that's really important to note is just because you have a lot of antibody floating around doesn't necessarily mean it's going to be problematic for the recipient. So if we have a patient that is incredibly sensitized, they have a ton of different HLA proteins, all that we really care about is, does our patient have antibody against that one specific donor's HLA proteins? If they don't, another anti-HLA antibody that's not specific to anything that donor has is just another antibody that we have floating around. So it's going to impact the graft no differently than an anti-flu or an anti-COVID antibody is going to. So if we look at the data on here, if we have a patient that has 100% CPRA but has no antibody against the donor, they respond no differently than if that patient was 0% and had absolutely no anti-HLA antibody. The only time we see a difference is in this red line here, where we know that before transplant, our patient has antibody that is specifically directed at one of those donor HLA proteins. If they don't have any reactivity against the donor HLA proteins, they're perfectly fine. So we always want to make sure that our patients before transplant either don't have any anti-donor HLA antibodies, or if we do, that we're able to identify that and be able to treat the patient once we perform that transplant. So as I said before we do a transplant, one of the most important things that we need to know is, does our patient have antibody against that one specific donor case? So in the case of our cross-match, we want to know one donor, one recipient. What's going to happen when we take that donor organ and put it into that specific recipient? So in this case, we take cells from the donor and serum from the recipient, and mix them together and see what happens. We have a couple of different ways that we can do this. We can use the flow cytometer, which is a little bit more specific. It only looks at binding, and it's kind of the most common way that we do cross matches at this point. There's the older method, the cytotoxic cross match, which uses a complement-dependent cytotoxicity method. It's less sensitive, but in some cases, we still use this because it ends up being a little bit more clinically relevant. So even though a patient may have antibody, is it an antibody that's necessarily going to attack and kill those donor cells? And in some cases, the flow cross-match may be too sensitive to identify whether or not that's the case. But instead, in a cytotoxic cross-match, if we mix them together and it causes the death of the cells, that's telling to us that this is an important antibody that may cause a problem. We've kind of moved on in our methodology on this. So instead of doing flow cross matches or cytotoxic cross matches on everybody, we've gotten more into other ways to do this. So part of the reason is there are lots of complications that you can have in a cross match. So there are patient-specific interferences with things like autoimmune disorders. There are therapeutic antibodies that can interfere with this testing, donor issues, timing. All of these things make it so that sometimes the physical cross-match may be problematic or may be prohibitive to use in a given situation. So what we've done is we've moved to something called a virtual cross-match. And a virtual cross-match is really kind of a prediction of what a flow cross-match would look like, or more importantly, just a way of gauging how much risk there is with putting a specific donor organ into a patient. So the way we do this is we take a patient's antibody screen that we get on a monthly or quarterly basis, and we get a donor offer on this patient. We say, okay, we have a donor offer for Mrs. Jones. This is her antibody profile, and this is the donor typing. So we get the donor typing from the OPO laboratory that performs the testing on the donor. We get this donor typing, and now what we're doing in the virtual cross-match is we're trying to see, does our patient have antibody against any of these donor proteins? So if we look at this, we see the donor A2 and the donor A3. Well, in this case, this patient has a fair amount of antibody against these two proteins. So this is likely going to be a positive virtual cross-match. In most circumstances, we would likely not move forward with this transplant because the likelihood is this patient would end up having rejection as a result. But if we have a different donor here with this typing down here, you can see that all of the beads representing those donor proteins are negative. This is likely a negative cross-match. This is a good donor to use for this recipient. Some are more complicated and some circumstances are going to be more complicated. So if we have this donor here, they may have a bunch of weaker antibodies that may or may not impact the donor organ itself. It may depend on how sick this patient is. They're a heart patient and they're going to die within the next couple of days if we don't get them a transplant. We may be more willing to take a chance on some of these weak antibodies and put an organ in there to make sure that they're okay. So in all of these cases, our final goal is to try and ensure that any donor organ that we're putting into our patients are going to be a safe and effective transplant for that recipient. So with that, just a quick summary that our HLA proteins make up our tissue type and are the targets of organ rejection. Rejection is mediated by both cells and antibodies. And in the laboratory, our job is screening for these antibodies to try and help reduce the incidence and severity of rejection, and that good pre-transplant antibody monitoring allows for not only accurate virtual or predicted cross-match results, but allows for safer transplants for our patients. So with that, I am happy to go through and answer any questions you may have.
Awesome. Thank you, Dr. Lalli, for that excellent presentation. I'm now going to open the floor for questions. So if you haven't already, go ahead and submit questions in the chat feature, and I will allow as many as time allows. But if we are unable to get to your question today, do not worry. We'll forward all the remaining questions to Dr. Lalli, and responses will be shared afterwards. You will also have another opportunity to submit questions through the webinar evaluation as well. So I'm going to go through what we have so far. The very first question is, why can't you just immunosuppress a recipient and transplant whatever you want?
So, wait, ask that one more time.
Why can't you just immunosuppress a recipient for a transplant, whenever you want?
Got it. So immunosuppression is not benign. There are a lot of problems that can come with it. So if you immunosuppress your immune system, it makes you more susceptible to infections. So, for example, one of the leading causes of graft failures that we have is through something called BK viremia. So there's a virus called BK that under normal circumstances, our immune response is able to keep at bay and doesn't cause problems. But when we immunosuppress our patients, sometimes these viruses, BK, CMV, EBV, can reactivate and actually cause a lot of problems. So immunosuppression itself isn't just some benign thing. It can cause, in some cases, more problems than it helps. So it's a very fine balance between preventing the rejection of the graft and allowing for the immune response to fight off these infections. So you need to be able to have that balance. And in some cases, the immunosuppression can actually cause the problems that are out there.
Interesting. And the next question is, do all organs require HLA matching and antibody monitoring?
They do not. It's actually quite a continuum that we have. So if you look at what happens inside of a graft or what the graft is actually doing, the functional portions of it are very different. So if we look at a kidney, for example, kidneys are generally the most prone to antibody-mediated injury. If you look at the business end of a kidney, those glomeruli, the tubules are really fine microstructures inside there. So a little bit of damage to those fine structures causes a lot of problems. So we need to be very careful about how much antibody a kidney deals with. The opposite end of the spectrum, if we look at the liver can take an absolute beating. So the hepatocytes within the liver are very regenerative. So even if you have a ton of antibody-mediated rejection inside of a liver graft, those cells are constantly turning over. They're basically saying, come on, bring it on. They're fine, to the point where in the laboratory, a lot of places don't even test liver patients for antibody. We just don't care because the liver can handle it. No reason to spend the extra time and money doing the testing if it really doesn't matter. And then hearts and lungs are somewhere in the middle of that. Hearts can take a little bit more of an antibody insult than a kidney or a lung can. And it all kind of depends on the patient, the quality of the graft, all of that kind of stuff. So certainly a big difference between the amount of antibody injury that a kidney can take and the liver.
That makes sense. Next question is, how far along does the pregnancy have to be in order for the mother to have an HLA proteins from the father?
Generally, it actually happens during the birth process where blood from the baby can actually enter. It's kind of the same situation as the Rh factor that we have. So the reason for having RhoGAM. Other than that, I don't know the biology well enough to know if it happens at any other point in time, if the anti-HLA antibodies form during, or what the time frame is on that one. I actually don't know the answer.
I'm going to do one more question. How does the allo stem cell transplant treat solid organ graft rejection?
So what I'm assuming the question is there's been an idea that if you do a stem cell transplant from the same donor that you're receiving the solid organ from, the idea is that those cells are not going to recognize the donor organ as being foreign, right? So it's from the same individual. So those donor immune cells that you're using in the allo stem cell transplant are going to look at that donor organ and say, yep, I recognize this. This is okay. So the idea with that is that you can induce tolerance in that recipient by giving them these donors, the donor immune cells. And in some cases it works, and in some cases it doesn't. It also adds some other complexity to it because you can induce things like graft versus host disease, where the transplanted donor immune cells can actually start attacking the other cells in the recipient and not the transplanted organ. So there's other complexities to it. And in some cases, even when you do this allo transplant, it still doesn't prevent the attack by those donors or from the cells against the specific organ itself. So it's certainly something that's in the experimental phases to see if there are ways that we can influence the immune response within the patient against the donor organ, but there's still a fair amount of study that needs to happen to perfect that.
Awesome. Well, I know we have a lot of questions in the Q&A, but we are unfortunately out of time. Thank you again, Dr. Lalli, for sharing your knowledge and expertise today with us. And thank you to everyone who has joined us on this sold-out webinar. As a reminder, please complete the evaluation that you'll receive by email later this week. And once it is completed, your CE certificate will be sent automatically. I also encourage you all to explore the Osh University for any additional educational resources and upcoming webinars. Thank you, everybody, and I hope you have a great rest of your day.